All right, welcome to part 2 of Unit 2. So we are again talking about the biological basis of behavior. Today what we're going to be talking about is neurons. And there's going to be basically two kind of parts. The first part is just going to be, what a neuron is and what the different pieces of it are and what they do. The second part is going to focus on neural impulses, which is actually how that information is going to be sent through the neuron itself. Okay? So the first kind of question is just, what are neurons? And basically what neurons are the simplest piece of our nervous system, of our information system. Okay? So they are going to be making up basically the majority of our nervous system, right? So we saw how in our nerves, right, those are basically bundles of these neurons. We have basically the same thing in our brains, right? Our brain is also mostly a bunch of neurons that are all interconnected to each other and kind of working together to send information in different ways. Now, neurons can look multiple different ways, as you can kind of see here. There's different types of neurons. We're really going to focus on this guy right here, which is basically just a standard neuron. Know that even different ones pretty much all function very similarly. There are some that will be a little bit different. But we're going to really focus on just kind of a standard neuron and apply that in general. Now, one other kind of piece to know that's kind of important for us, and we'll talk about it later as well with neuroplasticity, is that neurons are born and they do die as we kind of go through our life. So just like skin cells, right? We know that we create new skin cells as we go through our life. The same thing happens with neurons. We don't necessarily have the same neuron for our entire lives or anything like that. So this is just kind of our first really simple kind of diagram of a neuron, and there's all these different parts. We're going to talk about each of them specifically. Basically, we have our dendrite, we have our nucleus down here, right here. We have our cell body, which is around it. We have our axon, which is this kind of part right here. You can't see the whole thing, but if you imagine there's a tube kind of running through all of this, that's our axon. We have our myelin sheath, which are these kind of pill-looking things that are wrapped around it, and then it branches off again, and we have our axon terminals at the end here. Okay? So let's talk about each of these more specifically. Now, our first two parts, the cell body and the nucleus, should be pretty simple because they're basically function the exact same way a cell body works in any cell and a nucleus works in any cell, right? So the cell body is that kind of body part, right? If we go back a little bit to look at those first neurons I showed, right? Here's our cell body, here's our cell body, here's our cell body, right? In each one, there's a nucleus in the middle. Okay? So our cell body is going to keep the cell alive and is going to contain the nucleus, right? So just like any other cell, it's going to have, the nucleus in it. It's going to have basically all the stuff in there that has to do with basically making sure that there's energy in the cell, making sure the cell doesn't die, making sure it has energy to burn, right? All that kind of stuff is going to be in the cell body. Now, in addition, we also have the nucleus, right? The nucleus is going to do exactly what the nucleus always does in every cell. It's going to contain the DNA, right? It's going to have those blueprints for the cell itself in there. Now, the last piece on this first kind of image here is our dendrites. Now, our dendrites are kind of the first thing that makes neurons look very different than other cells. And you can kind of look at them, they kind of look like branches on a tree up here, right? And what they're doing is they are receiving information from other neurons, right? Or technically it could be sensory organs as well, right? So they're basically pulling information in, they're gathering information, generally in the form of neurotransmitters, right? They're gathering information and they're pulling it into that cell body, okay? So again, information is coming in, right, and it's going to this cell body. Our next piece is, again, our axon. So again, our axon is that kind of tube part. It runs all the way through this myelin sheath that's actually wrapped around it. And what the axon does is it is basically shooting the information from one part, which was our dendrites, which were over here before, shooting it out to our axon terminals on the other end. Okay? So it's shooting that information from one place to another. In addition, right, we have our axon terminals. Those are basically the ends right here on the opposite side. Now again, these are not the dendrites, these are the other side of it. And what it is going to do is then basically pass the information from the end of this neuron to the next neuron, right? So if you can imagine there's another neuron here, you can imagine there's dendrites, right, that are over here. It's basically sending this information to that next neuron. Okay. Our last piece is the myelin sheath, and again, these are these kind of green-looking things that are wrapped around. Just kind of ignore the red dot in the middle. That's something that we're just gonna ignore it. But these myelin sheaths. So what the myelin sheath is basically fatty tissue that helps increase, right, how quickly information is going to be shot through that neuron. Okay. It also helps protect the axon. Right. It also helps protect the axon of that neuron as well. So it's going to protect it, and then it's going to speed up how quickly information is shot from that, those dendrites, that cell body, through the axon to those axon terminals at the other end. Now, the next kind of thing we're going to talk about are what are called glial cells. So glial cells are not neurons, but they're cells that are very closely connected to them. So we're going to talk about them a little bit here. So basically the job of glial cells is to support for and care for these neurons, right? They want to make sure that the neurons have energy, that they're fed, that they're protected, right? That if there's like debris in the area, that is taken care of, right? That's all stuff that glial cells are going to do. Now again, there's a bunch of different kinds. You don't need to know what the different kinds are, but there are a bunch of different kinds of glial cells that have their own kind of jobs, right? So, and there's kind of a list right here, right? So things that glial cells will do are provide nutrients, as well as myelin, right? So you can see this one kind of creating that myelin sheath, guiding neural connections. So when we talk about neuroplasticity, we'll talk about how these connections actually change, and neurons basically search out and find each other and connect with each other. They mop up ions and neurotransmitters. So if there's extra stuff, again, extra things floating around, these glial cells are going to basically, make sure that they're not going to damage or create any problems for the neurons, right? In addition, right, they are involved in things like learning, thinking, and memory. We aren't like positive exactly all the things they do or how they're connected, but we know, again, partially because they're so connected to our neurons, that they do have a role to play in kind of all these different things that we do. All right, this is a good place to do kind of pause if you want to go through and do some of that coloring out of those coloring anatomy books, right? Again, labeling that neuron, and kind of coloring through all those different pieces. Or if you drew your own, you can do the same thing. But we're going to kind of continue on here and start talking about neural impulses, right? So neural impulses are how information is actually going to go from one neuron to the next. And you can kind of see a really simple image of it, right? This GIF down here of that information coming into the dendrites, going through that cell body, shooting down the axon, and then getting to our axon terminals at the other end. So the big thing here is you need to understand the steps of a neural impulse, right? You need to understand exactly what happens when those neurons will fire, when they won't fire, and how that's going to happen, okay? So the first step is basically there has to be some sort of stimulation of that neuron, okay? Now, this can come from kind of two main places. It can come from another neuron, or it can come from a sensory organ, right? So for example, let's say like you feel a cold breeze, right? That those sensory organs in your skin that are sensing temperature, right, are going to be basically excited, and they're going to then basically give that information to a neuron, and that neuron then is going to go through this neural impulse, okay? So it's basically going to be excited by either a sensory organ or another neuron, okay? Now, for that neuron to actually fire, for that information to actually be sent, right, it has to reach what is called the threshold, okay? Threshold is a really important term. It actually should probably be highlighted here, but threshold, right? So it has to reach, again, what we call threshold. Now, what is threshold, right? So basically what threshold is the minimum amount that an excitatory signal must exceed an inhibitory signal. Now we're going to talk about excitatory versus inhibitory signals a little bit later. Very simply, an excitatory signal is telling us to shoot, to fire, right? This impulse, an inhibitory signal is telling us not to fire. Okay? So basically, we have to have so much more excitatory than inhibitory. Okay? One way to think about it is kind of right here, right? So if you think about kind of these little fountains here, right? Basically, if you had one that was completely, like the water was only in the bottom, it wasn't in any of these pots, right? And you started it, right? The water would flow over, once it was, over this rim. It would flow into this first pot, right? And at first, it would just sit there, right? The pot would fill up, fill up, fill up, fill up, fill up. Eventually, it would pour over, right? And the same thing would happen here, right? It would have to fill up to a certain degree, and then it would pour over, right? The exact same thing kind of happens here. Okay, so the exact same thing is kind of what's happening here. So we have to reach this basically level before we're going to fire. Now, once that threshold has been reached, right, a neuron is going to fire, right? And when a neuron fires, it is called an action potential, right? An action potential. Again, this term should be highlighted. Sorry, I didn't highlight these for some reason. So it's going to have that action potential. Now, again, action potential is the actual firing of the neuron. Okay, it's kind of tricky because the word sounds like it might fire. Action potential means it is firing. Threshold has been reached and that impulse is being sent. Okay, so what is an action potential? Basically, what it is it's a brief electrical charge that's going to shoot down the neuron. Right, so you've probably heard that like our brain and our neurons function through electricity. Well, this is what that electricity is. Right, it's electrical information that's being sent down that axon of the neuron. Right, so again, it's starting in our cell body, it's being sent down the axon of our neuron. Eventually, it's going to reach our axon terminals. Okay. Now, exactly what the action potential looks like varies neuron to neuron. They can go anywhere, and you can see this at the bottom here, right? Anywhere from 2 to 200 miles per hour. Right. Now, it is important to know that this is actually relatively slow. Right, it's very quick for us as human beings, right? If you think about something going 200 miles per hour and it's only going from like your brain to your toes, that's very, very fast. But when we compare that with like electricity, right, in like wires or in a computer, it's actually 3 million times slower, right? Which again is kind of something that we can think about why computers or machinery is able to do things so much quicker, like certain kind of tasks, right? If it's just making, if it doesn't have to make a decision, right? If it's just kind of doing something, it's just getting information from here to there, it's actually much, much faster than we are, okay? So although it's going very quick, it's pretty slow compared to kind of other electrical currents. So let's get a little bit more in depth here, and this is where things can get a little complicated and a little confusing, right? But we have to kind of know what is actually happening in an action potential, right? Because neurons don't send electricity down themselves the way like a wire does necessarily, okay? So basically what's going to happen is there's the inside of a cell and the outside of a cell, right? The inside of the neuron and the outside of the neuron, right? And you can kind of see these two sections here in this image, right? Now, I want you to think of this as the axon, right? So the axon is this part right here. This red, right, kind of more red colored part is the inside of the axon. This blue part is the outside of the axon, okay? Now, inside of our bodies, and what these neurons are in is kind of like liquid, right? I don't know if that's maybe the best way to describe it, but there's basically stuff like floating around in between all these cells, right? It's not just like empty air, right? There's like neurons and ions and different chemicals and different minerals and vitamins and stuff like that are inside all of our cells in our bodies. So basically the inside of a neuron, right, has neurons are filled with negatively charged protein ions and positively charged potassium ions. Okay, so inside there are again negatively charged protein ions and positively charged potassium ions. Okay, now outside of the neurons, right, they are surrounded by positively charged sodium ions. Okay, So the outside is these positively charged sodium ions. Inside has both negative and positive, but if you kind of look here, you can see it's mostly negative inside, right? So inside is negative, outside is positive. Okay, so what happens is basically once a threshold is reached, what it does is it causes a gate to open up in our axon. So there's all these little gates, you can kind of see them here, right, that are basically in our axons, right, or are kind of opening up. They're like gates to a community or something like that, right? It's kind of like a gated community. So when we reach that threshold, it causes these gates to open, okay? Now, if you think about like magnets, right, and having positive charges and negative charges, right, what happens when you have a positive and negative charge near each other, right? They connect, they like shoot towards each other, right? So the way that works in this kind of form is basically when these gates open up, all of the stuff that's positively charged rushes in to the negatively charged inside of the neuron, right? So all this positive energy, this positive electricity, right, this positive charge is shooting into that neuron, okay? And what that's basically going to do, and I'm trying to move so that thing will get out of the way, right, and you can see it on the very bottom there, is it's basically going to cause this wave, because as that rushes in, it's going to basically also push out some of that, the positive charged, not the proteins, I'm sorry, the potassium ions, right, those positively charged potassium ions, right? So it's kind of pushing and pulling all this thing, and what it's going to cause is basically this wave of electricity to shoot down the neuron, right? And you can see in that little kind of chart there, right, you can see we're usually at negative 70, it's like, I believe it's millivolts, right? But as that positive charge rushes in, now we're going to have this positive electrical charge, and it's going to be pushed basically down that axon. Okay, so again, it's complex and it's kind of confusing, but we'll kind of go over a few ways to try and hopefully remember this a little bit better. But again, we've got positive on the outside, negative on the inside. Positive, when the gates open up, the positive stuff rushes in, right, which pushes some of that negative out, and it basically gets pushed down the axon. Okay. So again, you can kind of see it here in just kind of a more static form, right? So the first part A, right, is in response to a signal, the soma end of the axon becomes depolarized, right? The soma end is basically this where the cell body meets the axon. So right here, right at the top of the axon, it becomes depolarized right here, right? So all of a sudden, there's all this positive charge that just rushed in. Okay? Now as that happens, right, it's spread down, right, the axon, right? Because again, positive and negative are attracted to each other. So this stuff is rushing this way, right, to get to all this negative charge. Again, you can kind of think of magnets there. So the depolarization spreads down the axon, right? Again, you can see it moving down the axon. Now, while that's doing that, the first part, right, begins what's called repolarization. So basically, once this positive charge rushes in and starts moving down, right, there's a bunch of different things that are kind of happening here, but basically the cell starts evening itself out. It starts getting back to where it normally would. It's that resting, right? So it's polarized because the Na, right, because the protein, right, no, Na salt, sorry. The sodium, right, because the positive sodium channels are inactive and additional potassium channels have opened, right, the membrane cannot depolarize again, right? So basically there's like little pumps here, right, and because of the different charge that's happening, as this goes down, this stuff starts trying to settle back into where it normally is, and it basically is going to balance itself out. Now again, I'm trying to get that thing to go out of the way, there we go, right? And so again, that's going to basically just continue to happen. Now, it's showing this in like chunks, like it happens here, then it happens here, it happens here. It's more of like, again, like a wave, right? So it's basically like the stuff is rushing in, and as it's rushing in, it's being basically pushed right down, rushing in, and then it's starting to be pushed out behind it, okay? you can kind of think of it like water as well. If you've been to like a wave pool, right? When that wave basically gets, right, that giant wave washes over, right, it like sucks all of that kind of water with it, but then, right, it kind of comes back and it settles back down to normal, right? It starts filling that space basically. Okay, so what happens afterwards, right? So once that action potential happens, so once it's fired, right? So again, we're sitting here, right? Our positive sodium is rushing in. We get that positive charge, right? This is the action potential right here. It's firing, right? What happens afterwards? So basically, it's going to go through what is called the refractory period, right? Right here again. Again, important term, should be highlighted. I'm sorry I did not, right? Goes through this refractory period. Now, what the refractory period is basically where the cell cannot fire, right? Now, this is very quick. It's right here, a fraction of the blink of an eye, right? It's very, very fast. It's not a long period of time, but there's a short period of time where it cannot fire. And what's basically happening is there's pumps, again, in there, there's biological pumps that are bringing it back to its resting state. So usually what happens, right, we have this, again, action potential, right? We have all this positive charge. That positive charge is being sent. Then as that kind of charge gets towards the end, it starts coming back down, so it becomes more and more negative, right? We're pushing out the potassium, right? Oops, I'm sorry, I didn't mean to click there, right? So we're repolarizing, right? Now, what happens is usually there's this little phase which is called hyperpolarization, which basically like we get a little bit too negative and then it balances itself out. Okay, now once we're balanced again, once we get back down to negative 70, now we can fire. Okay, so there's this chunk here where we can't fire anymore because basically all of the energy, the electricity that pushes that through is been used, right? So it has to rebalance itself. Okay, now neurons, right, and this is kind of explaining that as well, have an all or nothing response. Right? So what that means is they either will fire 100% or they don't. Okay? So and that's why I have this little GIF here. I kind of like to think about it like firing a gun. Right? When you are basically like pulling the trigger, right? If you pull the trigger halfway, the gun doesn't shoot. Right? It doesn't do anything. Right? Nothing happens until you pull it past that point that's going to send the shot. Now, once you pull it past that point, that's it, right? It's going to fire, okay? Now, if you pull it super hard, it doesn't make a difference. The bullet's not going to fly any faster because you pulled the trigger harder, or if you pull it soft, as long as you hit that threshold, it's going to fire. Neurons are the same way, right? It doesn't matter if there's tons of excitatory, right? If it reaches threshold, no matter how much more past that threshold it goes, it fires with the exact same strength every single time, okay? In addition, kind of like same thing with a gun, right? And you can kind of see it here. There's that moment where the gun can't fire, right? A weapon can only fire so quickly, right? Even, like really, really like high rate of fire machine guns, there's small gaps in between everything, right? And you can see that, right? So once it fires, there's this short moment, just like our refractory period, where the gun cannot fire. And you could pull the trigger 1,000,000 times if you could, do it that fast, and it wouldn't do anything because it's not ready to fire again. Okay, again, our neurons are going to function that same way. So just to kind of go over it really quickly one more time, right? We have our threshold, so information comes in. If we reach this threshold, this level, we then have our action potential. Our action potential is that electricity. So the electricity is shot down our neuron, right? It's basically pushed. So that was positive sodium ions rush in, right, and it causes this electricity to get shot down our neuron, right, push down that neuron. As it's being pushed, right, it's also then basically repolarizing, where it's going to go through that short refractory period before it can then fire again. Okay, so that is our neurons, right, and again, this is the basic building blocks of everything else in our nervous system. Okay, so go ahead, please do a quick summary. Please go and do any of those like coloring books or labeling pieces that you can, just to kind of make sure we feel like kind of understand these pieces really good. Next time, the next video, you guys will talk about neurotransmitters, where we'll actually start to see, okay, how does one neuron communicate with another neuron? So far, we've basically only talked about what happens within one single neuron on its own. What happens now when we start connecting?